In this presentation, we are going to discuss post-infectious irritable bowel syndrome, or PI-IBS. Post-infectious irritable bowel syndrome is the development of irritable bowel syndrome following infection with gastroenteritis, with more recent definitions being the development of IBS after acute infectious gastroenteritis. Although PI-IBS was first identified during World War II, research continues to be conducted on this illness as scientists search for determinants and ways to treat symptoms. Recent studies have suggested a genetic predisposition, and scientists are looking at therapies aimed at mechanisms such as immune activation and serotonin excess in order to treat this illness. PI-IBS is significant as it is proving itself to be a key player in learning more about the pathophysiology of all IBS. In terms of prognosis, there has been a reported slow decline in the occurrence of PI-IBS since its initial diagnosis. A 6-year follow-up study of post-infectious irritable bowel syndrome revealed that 43% of patients had recovered. A study following an outbreak of waterborne gastroenteritis from an E. coli infection in Walkerton, Ontario, revealed that the prevalence of PI-IBS went from 28% to 15.4% after the course of 8 years. Thus, given the data provided by these two studies, it is shown that PI-IBS symptoms will eventually decrease, but it is also important to remember that these symptoms may persevere for years on end. The prognosis is one of improvement, yes, and the chances of developing a new, similar condition are unlikely. However, symptoms may not immediately ease up.
Mechanism, and we'll start with inflammation. Enteric bacterial pathogens can cause secretion, inflammation, and epithelium damage via the secretion of cytotoxins. This can lead to macroscopic changes such as edema, hemorrhagic spots, ulcers, and a marked increase in gut permeability. During infection with viral gastroenteritis, there is a reduction in villous surface area and an increase in intraepithelial lymphocytes. Following protozoan infection, there is also a reduction in villous surface area, along with an increase in plasma cells and leukocytes in the lamina propria. IL-1 beta and IL-6 expression is increased in IBS patients, even 3 months post-infection. Now for the serotonin and mast cells. EC cells respond to nutrients and bacterial products in the lumen by secreting molecules that activate enteric nerves. EC cells have granules that contain 5-HT, or serotonin, which plays an important role in the motility and secretion of the gut, including in response to bacterial toxins. EC hyperplasia is associated with increased release of serotonin, leading to diarrhea in IBS. An increase in mucosal mast cells has been found in IBS patients. Mediators released from these mast cells may contribute to the increased severity of visceral pain. Now the altered microbiome. The microbiota normally helps protect the host from pathogens by producing SCFA, or short-chain fatty acids, which have antimicrobial properties. During acute bacterial enteritis, the inflammatory response inhibits the microbiota, reducing production of SCFA and increasing pH. These changes decreased diversity of the microbiota and promoted growth of specific bacterial groups, an effect that was found to last for at least 8 weeks.
In terms of risk factors, there's a genetic component. IBS shows a familial tendency, and an early study on monozygotic and dizygotic twins in Australia reported a heritability of 57%, but later studies have suggested a weaker effect. It was shown that IBS was greater in monozygotic twins than dizygotic twins. Having a mother with IBS was a stronger predictor than having a dizygotic twin with IBS. Studies of a single nucleotide polymorphisms, SNPs, also support a genetic influence. A greater proportion of IBS patients are heterozygous for the negative 308 GA SNP, which are higher producers of TNF alpha, tumor necrosis factor alpha. Is an inflammatory cytokine produced by macrophages in monocytes during acute inflammation. Possession of both a higher production of TNF-alpha SNP -308 and lower producer of IL-10 -1082 A allele was also more prevalent in IBS patients versus controls. More recently, the G allele of SNP RS42633839 in the TNF-SF15 gene, which has also been associated with Crohn's disease, has been demonstrated to increase the risk of IBS. And this is used from studies of patients in Sweden and the United States. A closely related SNP in the TNF-SF15 gene has also been shown to increase the risk of IBS-D, which is diarrhea, but not IBS with constipation, IBS-C. The same study has also found an increase in prevalence of the TNF-alpha SNP RS 1800629 genotype GA compared with the GG in PI-IBS. Further SNPs associated with PI-IBS have recently been reported. There have been 3 gene regions identified where SNPs increase the risk of PI-IBS, namely cadherin-1, IL-6, and toll-like receptor 9. Cadherin-1, coding for E-cadherin, is a tight junction glycoprotein which influences intestinal barrier function. Toll-like receptor 9 is the receptor for unmethylated CpG dinucleotides, which are a marker of bacterial DNA and represent the pathway whereby bacteria activate innate immunity. IL-6 is an inflammatory cytokine activated by infection. The studies conducted to reveal this information are good, but they do have relatively small sample sizes, which is a limitation. Other risk factors include physical and psychosocial. Several studies confirm that high stress and anxiety levels, adverse life events preceding 3 months, and depression all increase the risk of developing PI-IBS. A high level of T lymphocyte and enterochromaffin EC cell numbers also increase the risk. Other factors include age and smoking. Being older than 60 years old protects against PI-IBS, possibly because of the declining immune response with aging, while smoking increases the risk, but the mechanism is unclear. The underlying mechanisms whereby these psychological factors increase the risk of PI-IBS are unclear as well. Scientists have hypothesized as to why using animal studies. Studies show that the corticotropin releasing factor, CRF, is an important mediator of the stress response. It acts via the hypothalamic pituitary adrenal axis in animal models of PI-IBS. CRF acting via CRF1 receptors mediates the stimulation of colonic motility by various stressors, and CRF in stress enhances abdominal pain through the central CRF pathway and activation of mast cells in rats. CRF may also be pro-inflammatory, stimulating lymphocyte proliferation by increasing IL-2 receptor expression and enhancing production of both IL-1 and IL-2, as well as enhancing cytokine release in response to endotoxin. Stress also aggravates chemically induced colitis. Thus, by enhancing the local inflammatory response to infection, stress may increase the risk of PI-IBS. So basically, when you get stressed, your body tries to mediate the response and causes inflammation in the process. This has been examined in more detail recently using an animal model of PI-IBS. Rats infected with Citrobacter rodentium, a mouse-specific pathogen used to model infection of E. coli, who were exposed to chronic water avoidance stress, showed increased level of corticosterone and epinephrine levels and an increase in pain and rectal distension. They assessed this by recordings and measuring nerve responses. They also found an increase in tissue proteases and homogenates of the colon of infected mice, most likely from activated mast cells, which induced hyperexcitability in colonic dorsal root ganglia cells. Human studies have also shown activation of mast cells. They did this by immersing their hands in cold water to induce pain and sympathetic activation, which raised the pulse and blood pressure. This led to the release of mast cells produced, such as histamine and tryptase, into the lumen of the small bowel, providing a possible mechanism for how stress could increase human small bowel secretion and motility, leading to an accelerated transit, which is a characteristic of IBS-D diarrhea. The same group have also shown evidence of increased numbers of mast cells in anxious IBS-D patients associated with increased release of tryptase from biopsies, a finding recently replicated by Foley et al. Bacterial toxins play an important part in development of intestinal inflammation during infection too. C. jejuni infection is one of the most common causes of infectious gastroenteritis, leading to PIIBS in 9 to 13% of cases. Patients infected with a strain that secretes toxins can cause elongation of human epithelial cells and have an increased risk of developing a long-term change in bowel habits. Other bacterial pathogens that cause marked gut inflammation and have been associated with PIIBS include enterotoxigenic E. coli, the Shiga toxin producing E. coli O147, especially when combined with C. jejuni, Shigella, and S. enteritidis.
Regarding management of PIIBS, the illness should be handled on a case-by-case basis according to the severity of symptoms. Treatment should be in accordance with the individual symptoms, such as abdominal pain, anxiety, or diarrhea. Currently, there is not a defined treatment for PIIBS, but recent studies have brought out potential fighters against the battle of PIIBS. Pilot studies using Madelene, an anti-inflammatory agent, showed results indicating a reduction in mast cell numbers and improvement in abdominal discomfort, as well as an altered gut microbiome. However, it is important to note that this study was underpowered and does not serve as a surefire treatment yet, but deserves further research using a greater pool. Ondansetron, an antiemetic serotonin blocker, also has potential as a treatment as it has a high safety profile and a randomized placebo trial published by the Journal of Neurogastroenterology showed a 50% reduction of symptoms in 70% of participants on this particular drug. Newer approaches are looking into the inhibition of serotonin synthesis in order to improve the symptoms of PIIBS. There is still a lot of potential research to be done, and these pilot studies are lighting the way to a pinpoint treatment for PIIBS.